Well, I'm back, and so are you, for another and probably last installment of the Empowerment Project. So today, what I'm going to go through with you is ordination, and we're going to have a lecture, and we're going to have a set of two R exercises that should, in total, give you some good practice in ordination. So let me just get things set here. Yep. Good. There we go. Okay. So I began last time talking to you a little bit about ordination, and actually, I in that multivariate approaches lecture, which you might not have watched, right? Because all of these are elective. By the way, there are no points involved in this. This is 100% just trying to help students who might be interested in doing ordination in the future. It does not relate to points in the class. It's empowerment only. This is empowerment only. And you may have skipped other things, but I talked about it or introduced it in the multivariate approaches lecture. So I am going to oh, sorry, I lost that. I am going to I am going to repeat some of the things I said before. I don't know why I did that. Biological phenomenon, though, are very often multivariate in nature. ANOVAs and regression and simple linear regression and correlation are just one variable compared to another variable. And oftentimes we have a very different situation, and we have many variables that we're trying to analyze at once. We must consider them all simultaneously before we're going to understand the system. A classic example of this is a species by plot matrix, which you can see right here. And you can imagine, for example, if we wanted to compare animals or insect or plant communities between a burned and unburned grassland, we might establish plots like this. Then we might measure microbes or plants or animals or insects. Insects are animals, but a certain kind of animal, insects. And or you might imagine samples of like microbial samples, gut microbiota or whatever, and we have some samples, and in the samples is a complex data set. For example, like this, where you've got some samples, this is like plots, and this is a species, this is a species, this is a species, this is the first three letters of the genus and first three letters of the species, and this is some measure of abundance for those. And you may have a lot of those. In this one, we've got like maybe 20 or something here, but this can go on this way for a long distance. And when you start to ask the question, how did fire influence this ecosystem at a community level, it's not that easy to analyze. You're going to have to use a multivariate approach. We may also be interested in environmental parameters measured in the same plots. So these are actually the same plots as I presented before. So you might have plots that have some species in them. Then also about those plots, we know other things like the phosphorus concentration or cation exchange capacity or something like that. Just zoom in here. Okay. So the statistical approaches we've used in class so far just can't handle this. It's not going to work. Multiple regression, if you paid attention to that, well, you could use multiple regression for some piece of this, but you're not really going to be able to answer the question sort of biologically, does fire influence prairie ecosystem, insect communities, for example, using those approaches? We're going to have to try to take a different approach. Similarly, if you sample microbial communities, what you're destined to come back with is some sample that contains a bunch of different organisms. Precisely what those organisms are, you don't even know. You might use operational taxon unit or some other thing, and you say, we've got these taxa present when we put the antibiotic into the plate. We've got this set of taxa present, and then the other plate we've got this other set. They're not the same. But what's happening in the plate is there's a bunch of different species that are affected all at once. It's not just maybe you're not interested in one particular species, but you're interested in the microbial community. If you're interested in the microbial community, then you have to ask a question at the community scale, and a t-test is not going to do this for you. You're going to have to do something else. Okay. Okay, so ordination is a family of analysis techniques that uses complex information about a set of samples to organize and display information. In ordination, samples are compared using some measurement the scientist has made. Often this is, as I mentioned, a plot or some sample, and the measurement we've made is taxa or species. Okay. Like, for example, herbaceous plants in a forest. Ordination puts the samples in order based on, in a two-dimensional space, based on the composition of the samples, okay? And, yep, and those are most oftentimes, the composition of the sample most oftentimes is some species or some other kind of measurement of the biological organisms present. Could be genera, etc. Now, just a very, very brief history of this technique, because some people don't really understand this, even though it's quite popular. The first use of ordination was by Bray and Curtis in 1957, and these were ecologists, and they were just trying to organize plots based on species composition in the plots, forest species composition. These are tree species, actually, and they called this polar ordination. And they just say, I'm going to put the plots that are most dissimilar farther apart. Dissimilar in terms of the species composition, and we just order them and make a line. Polar, or that's where the word ordination comes from. Order. We're ordering these. We're ordering them based on the species composition. It's worth pointing out that this was an ecological. It was invented by ecologists. That's all, because they were studying forests, and forests are complex. They've got lots of different species. Now, that kind of technique is oftentimes used by people who use different kind of tools, like microbial tools today, but it started with ecologists. And then beyond Bray Curtis, which was just like a line, and actually they developed this distance measure, which we're going to use a little bit later actually in this session. A whole bunch of things followed. 1957 was Bray Curtis, but then principal components analysis came along, a thing called reciprocal averaging and detritic correspondence analysis. There's been all kinds of other sorts of analysis approaches. What are popular now? We're not going to address all of those. And there's been, those are 4 listed there, here. There's probably 6 more we could talk about. And then more recently, these are the most popular ones: canonical correspondence analysis, principal components analysis, which is the same as above, and a thing called non-metric multidimensional scaling, which is NMS or NMDS. Okay, so canonical correspondence analysis is often used to simultaneously analyze community and environmental data. So if you have an environmental matrix and you want to analyze it at the same time as the community matrix, you use canonical correspondence analysis. Pause. Okay, principal components analysis is often used to distill complex variables into one or a few axes, or subsequently used for some other purpose. PCA is a way to take complex information and create, like, oh, here's principal component axis 1, and now we're going to run a regression with that against some other thing, for example. Non-metric multidimensional scaling in NMS or NMDS is often used to analyze information about taxa that have been collected in plots. This is very commonly used now. We'll do an exercise in R using NMDS. So I pause there because I want to say, for each of those things, and as with any advanced technique, if you're going to use one of these in your research, you need to spend time reading about it and dig into the technical details. This lecture will present you only with doors to walk through. Those words I just said about canonical correspondence analysis, principal components analysis, are insufficient. Okay, it's not okay. If you want to use it, you need to read about it, spend some time, dig in. We're just providing you with doors here for you to walk through, however you'd like to. Okay. Modern applications of ordination almost end up with a graph like this. You've got some axis here, you've got some axis here, and each of these is a plot or some other sample. So this is a sample. The location of the sample in this space is determined by the composition of the sample, oftentimes species, or taxa in a microbial sample. Oh, the gut microbiota. And then it's arrayed in this two-dimensional space in that way. What's really important for you to think about is you're often working with two axes, or you're presenting two axes like this. But ordination, modern techniques like NMDS, almost always are three axes. So you've got 1, 2, 3 like this. And you're really working with a cloud of points, and you need to think about that from that perspective. This is a cloud of points, and it gets projected onto the 2D surface, and it looks like this. This is a goofy, I got off a website. A light shining through a cloud is going to project onto and here, this cloud here, I'll put that there. It's kind of ridiculous. But the main idea, if I can just get this image to stick in your head, if you're going to ever use ordination, which some of you will, I almost guarantee somebody in this class is going to use this later if you stay in science. So of course, I also understand since this is an elective lecture, and it's the last one, it's got no points. It's quite possible no one will ever watch this. So cheers to no one. Slancha. If someone is watching it, let me tell you that I think somebody in the class, whether they watch this or not, is going to use ordination later. And if you can keep this cloud image in your mind, this very likely can help you in the future. Because you might forget or not pay attention to whether or not which axes you're presenting. And NMDS doesn't necessarily give you the two most explanatory axes when it first projects your plot. There's a process by which you need to ascertain that. We're not going to actually even do that in class today in my exercise, but I want to tell you that because you need to think about it later. Okay, so ordination puts samples in order based on their similarity. Samples are plotted in a two-dimensional space where nearness of the points in that space represents how similar those samples are to one another based on the measured parameters of the sample, which is often species. These points would be more similar to each other than they are to these points. By the way, I'm somewhat reading more on the slide. In the class, I don't do that, but in case somebody has a sound problem, I try to type a lot more on these slides. So if that's bothering you, sorry. But the point is these points or samples are more similar than these. That's what the NMDS is telling you. Okay. And they're arrayed based on some measure of abundance in the sample. This is a species. Here's some measure of abundance. This is the plot. Okay. So ordination puts samples in order based on similarity. The plots are arrayed in space. Here are dominant species in the sample. So this is, I think, the most important species in any of these particular samples, or this is arrayed in that way. So DIO represents the most important species. In this area, EPI there, UR down here. So this is the species space, and this is just a species. And these are the samples, and this is how they're arrayed. This can give you some good information, and we'll chase this down in the example I'm going to present to you. Axes are sometimes meaningful, and some techniques in the ordination, it will have the first axis explain the most variation, and the second axis explain the next most. So the axis can be quite important, and the axis can represent something important in your study. For example, this is actually real in the data. This is from a long time ago, an ordination I ran. But this was a gradient from this species to this species. So that was strongly correlated, as you can see, with axis 1. In effect, axis 1 represented a gradient across these species. It was a moisture gradient, actually, is what it represented. These are dry site species. These are more wet site species. So something to think about. In ordination, you may have more than two axes. You could have a third axis. I showed you a 3D diagram of that already. So here's an example of a real ordination I did with data a long time ago. This is axis 2 and axis 3. Now, why would I present you with axis 2 and 3? It's a 3D plot. This is NMDS, and actually in this particular analysis, axis 2 and 3 had the most explanatory power. That's what should be presented. So you don't have to chase that down if you do NMDS in the future. Just listen to those words, and then later you can chase it. So this is the plots. This is a plot. This is its location. This is the name of the plot, max EU-2. These are the species. So this little cross represents where this species is. And these are the first 3 letters in of the genus and last 3 letters of the species. This is Lysomachia tonsa, which is a wildflower, for example. You can imagine it like this. These being dominated by pines and these being dominated by maples, and there being some kind of gradient in the species composition like this. This is the purpose of NMDS, is to array the plots based on some compositional information, and perhaps pines versus maples is the dominant thing that drives composition. So you could get something like this. If you were studying these plots and you had a priori or a hypothesis about pines and maples and it turned out like this, you'd be happy. Say, oh, look, I've got a visual representation of the community organization based on the dominance of these species. That would be exciting. Okay, there are many different ordination techniques. You should read about them. I already mentioned these 3. If you're going to decide to use one of those 3. Each of these ordinations and all ordinations are associated with statistics that can tell you how confident you are. They're not going to be p-value and hypothesis testing per se. That's one thing that in effect, ordination is leaving the realm of pure hypothesis testing and returning kind of into the realm of exploratory data analysis. You can represent your confidence, how confident you are in a diagram, but you can't necessarily have a null hypothesis of no pattern and then test it with a p-value. There's some sort of ways that people are developing that you can sort of artificially do that, but generally speaking, ordination was not set up in that way. But there are statistics that can help tell you how confident you can be that the pattern is real. You're going to have to read these different techniques to be sure that you report the correct things. Oh, here's an exciting thing. Some ordination techniques allow for an overlay of vectors. This is a vector. These are environmental parameters. So if you've measured, remember I showed you that second matrix, the environmental matrix. You can sometimes portray or display the environmental matrix on your data set, on your ordination diagram, like this. And that could give you some good information about, what's happening in the plots. It can help you explain or understand the pattern. Here we've got 3 different communities, these open circles, these gray boxes, and these triangles. And we've got these vectors, and the vectors represent environmental variables. So again, we can imagine here if we had these pines and maples overlaid on there, that you wouldn't have that overlaid on your plots. That's just like for explanatory or pedagogical reasons, I have that there. But you can imagine that if you had something set up like this, you might be able to say, high light conditions, this is summer light. So the way the vector is pointing, that means increasing of whatever that parameter is. So this is summer light. This thing increasing summer light this way. We measured light in the forest, literally. And H ion concentration right here. This is hydrogen ion concentration. More highly concentrated hydrogen ions in the soil means lower pH. Biology. That's associated with favorable conditions for pine trees. While if you head down this way, and by the way, this would be the negative of that. This would be, so H ion concentration increases, pH goes down, that's more acidic conditions. Down this way with H ion concentration, this is more basic conditions because we're going against that axis, and this is going to be darker or shadier. So shadier, more basic soils with more calcium, you've got maples, more light and higher ion concentration that results in acidic conditions, or where you have pines. That would be the kind of the notion here. Now, some techniques also allow for the overlay of ellipses. A lot of ordinations have ellipses like this. This is especially if you have a real smeary data set, like it doesn't look like anything. You're like, I'm going to put an ellipses on there, ellipsis, so I can see what's going on. That's fair. Okay, that's fair. It's not cheating to put ellipses on the diagram, although I'm a little shy about it. And in my own work, I've never drawn ellipses on my figures. But you might be able to draw it like this and go, oh, look, these ellipses are not overlapping. In the maple community and pine community, or pine dominated plots, and the maple dominated plots are separated. Look at this. Okay, now let's move over to R and run some practice ordinations. I would urge you to listen to some lo-fi hip hop or other music. I can tell you, I'm serious, man. Good music makes coding more fun. If you learn nothing else from my class, that would be a good thing. So move over to R, please. Now, okay. Ready? Okay, this bit of code is ordination, or this ordination class script is borrowed and then modified to some extent from a script written by Master Eric Bradley Borth. Master Borth is an alumni from the class, and he actually wrote this script and presented it in class a while back. Remember, I told you that what oftentimes happens is students get really excited and start doing their own stuff. And so this is Eric's, not entirely, because I had to borrow and modify it a lot for myself to be able to present it to you. So it's kind of co-authored, but generally speaking, I give all the credit to Eric Bradley Borth for this one. Here's a picture of him. I'll pull him up as we go along. Not only is this person good at coding, he's also very good at tree climbing. Those are not often correlated traits, by the way, tree climbing and coding. But this is one particular person where that's true of. So Eric Borth says, check out this tutorial, this NMDS tutorial, and I can show you, I think I've got that here. Yep, this is a tutorial. This is a really nice tutorial that this person wrote that Eric kind of borrowed, and then I borrowed from that. Now we're going to install some packages. I've spoken to you already about packages in various sessions. So we're just going to install packages like this. I've already done that, so it's not going to work in my case. But if you just run your code like this, type the code, run the code, it'll install that package. It'll ask for a crayon mirror. If you haven't done this before, we skip the other ones. Just this is the way it works. You just run that. You can also run it with this blank, and it'll just ask you which package you want. By the way, I looked the other day. There are 10,000 packages. More than 10,000. 10,000. So you can do a package for everything. So we want to do this one, and then once you've got it installed, it's there forever, and you just Control R to turn that package on, and you call it library, and then whatever the name of it is. And then we can install this one, VEGAN. Oops, it's already done. So it's going to say, oh, no. It's asking me to do it. That's exciting. I've already done that, though, I think. So yeah, it's already in use. So that didn't make sense. But you get to see the crayon mirror thing pop up. So I'm going to turn it on. Now, before we go further, I want to tell you that this package vegan or vegan is extremely well known and often used. So let's go to Google Oh, there we go. And I'm going to type in here because I just want to show this to you. Vegan package. Didn't spell it right, so that's still fine. Here's this vegan package crayon. You click on that, it's going to pull up this PDF. This PDF is describing the package. Now, there's 10,000 packages, I understand. Every package has something like this. This gives you a notion of how deep this is. It's deep, 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 deeper than the ocean. Okay. This is a very popular one. It's an awesome package. These people spent a lot of time. They generated something that's absolutely awesome. It's got all kinds of stuff. You can just see all the things over here. We're going to use some of those things. We're going to use ordplot and such, ordtope, ordhull. We're going to use some of these commands in just a minute. I just wanted to show you this package because if you were going to use it, you can dive into it like this. And it has extensive explanations of all these things. I can't explain all of these things to you, but this gives you a notion right here of all the stuff you could possibly do with ordi ellipses and ordhull, which we're going to use a little bit later, and all of the details. Look at this. The details are there. I can't give you the details here. I don't know the details off the top of my head, but you can chase them down this way. And that's true for all the packages. All the packages have this information, and there's 10,000 of them. So R is endless. R virtually endless. Okay, so that's just one package. So now the other thing Eric Borth did, here we go. Where'd he go? There he goes. The other thing, I don't keep him there, I can't. The other thing Eric Borth did, which was really sort of nifty, and I don't really understand this completely, but he generated, and he got it from that, I think he got the notion from this thing, this tutorial, to actually to use R to create a random data set. So he wrote the matrix, the community matrix we're going to use. Here's the community matrix. There's 10 communities, there's all these species. They didn't exist before this code right here. So this is something for those of you who are super sharp in the class and are interested, excited about coding. Check that out. See if you can figure out how that thing works. Don't ask me, yo. I don't know. I haven't figured it out, at least. It looks like this is a with replacement. This is some kind of randomization piece right here, and it's saying make 10 rows, use community. This is actually not that complicated, so you can understand the product pretty well. And now we're going to make two groups. So type the code, run the code. You'll know it works when you display the code like this, or display the data set, and you've got it there, like this. And now we're going to make two groups of colors for the communities, red and blue, just like this. We're going to use this later just for display purposes. You wouldn't necessarily do this if you're doing NMDS. So let's see what Eric Bradley Borth had to say about what's going on here. This is good, because he wrote this bit right here too. I think this is good. What is NMDS really? Eric wrote this. It is a way to look at the similarities and differences between groups using many columns of information. So we have this nice community matrix, cleverly named community matrix, which we just named it right here. And we would like to see how these communities relate to one another. The first thing we might think of is making a scatter plot, but only two axes. So let's plot species 1 and species 2. So this is the plot Eric Borth wrote for this exercise. It's a bit complicated. I don't really understand why he made it so complicated, but we'll follow along. Here we go. This is easier for you probably, since I've been doing this tab thing. Here's the plot. So see if you can generate this plot right here. type the code, run the code. Plot looks like this. That was good. That was good. Now we can show even more of the similarities and differences between the communities if we add another axis for species 3. So this is his code, Eric Borth's code for a 3D plot. Like this. Bingo. Species 3, species 1, species 2. Scatterplot 3D is this package. If you don't have this package installed, this is not going to work, y'all. So get that package installed and see if you can make this plot. Got it? Okay. So interesting, right? And by the way, if I were you, I would borrow this. Here's something I should say. That's just like a philosophical piece. And I should have said it in the first day of class, but I said this multiple times, that R is an egalitarian sandbox. Now, part of what I want to say about that is what you need to get good at, and what you have to be comfortable with are two things. One is borrowing people's code and just running with it. I mean, that's what we do all the time. So this is Eric Borth's code. We're running with it here. You should take this code and run with it. If you've got something to do, use it for something totally different than this. This is what R is all about. Okay. You come up with your own way of using people's code. So you need to be willing to just borrow code. And the other thing is, you need to be willing to trial and error. If you don't know what's going on, like, oh, I don't know what type H means. Personally, I don't. So what about I? Let's just try it with I. I don't know what's going to happen. They went away completely. Okay. What about O? Does it have to be type H? Wow, look at that. That's pretty awesome. What about P? I have no idea. Here's the point. This is just ridiculousness. Oh, that's interesting. Here's what you need to be able to do and be willing to do. Try stuff. Just type stuff in there. You will not break it. It will work. Colors equals colors. We define colors up here. Anyway, those are the things I want you to say. I want you to buy into this. And part of what I want you to be willing to do is take code and just mess with it. So, and I will post all of my class code at the end of the class. You should download it. You've got your own kind of library that can get you started going different ways. Okay, I'll stop now. Okay, back to Eric Borth's script here. Okay, now we're getting closer to showing the variation because we have more than one species here. What about adding a fourth axis to display species 4 and a fifth axis to display species 5? Oh, hold on, problem. We are bound by our we are simple mortals bound by our three-dimensional universe. Graphing with more axes to show variation is not possible. Here we go. This is for your ease of reading. We're simple mortals bound by our three-dimensional universe. We can't simply keep graphing more axes to show the variation. However, it is mathematically possible to look at the variation in many dimensions all at once and then display the variation in just 2 dimensions. That's what NMDS does. That's what we're going to do right now. Okay, so now we're going to run an NMDS. Let me just run it here quickly. This is how we do it. Now, this is the code to run NMDS. So we write example underscore NMDS, which is, I know you want to see that. This is irritating what I'm doing. We've already, oh, wait a minute. Equals, so we're defining it here, equals community matrix, which we did up above. This is our community by species matrix. K is 2. This is a number of dimensions. So we're saying we want this to be 2 axes. A lot of times what we're going to do in NMDS is have 3 axes. But in the meta NMDS example, you say K equals and say how many axes you want it to generate. So type the code, run the code, and you're going to see this thing run over here where it goes multiple runs. This is the thing about NMDS. What it does is it goes run, 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 run. It runs through, and this is stress. See this right here? And what you do in NMDS is you want to minimize the stress. Stress, it ended up with 16.5. That stopped there. So we can do it again together here. Eric Orth had us do it twice. I don't know why. Take a look at the data. Here's a community matrix. Run the NMDS. This is the NMDS code right here. Now, we can plot the stress this way. Now, I've been reading a bit about this, and you'd have to spend some time reading about it yourself. Observe this similarity in an ordination distance. The idea was that if the points are close to the line, then it's good, but you're going to have to read about this. If you're going to use this for your research, you're going to have to dig into this and read about R squared, this non-metric fit and linear fit, and what's going on with this. But that stress plot is part of what you want to report in your NMDS, and then we'll create the plot here. So this is the outcome of this analysis report, or this analysis. This is kind of the thing you would report. You'd use this, generate this diagram. That's the ordination itself. Now, type the code, run the code. Okay. Now we're going to use this ordiplot function in the Vegan package. So here's ordiplot, which is kind of the same thing we just had. And now this plots species onto the ordination diagram this way. This is an orditorp, orditorp, like that. Type the code, run the code. Okay. Now that's the species. Now this plot, the sites on. Now remember, in this particular one, we had, and we can go back and look at the data set, community matrix. Instead of plots that was outlined as community, which is kind of weird. In any case, here you go. So the plots, samples, or sites onto the ordination diagram. So now we've got a diagram that has the species and the plots. Okay. Type the code, run the code. Okay. Let's continue. So now we're going to explore some other graphing options. This is going to be exciting. Let's do ordiplot again. Here it is. Here's just a plot. When you type this thing, type equals n, it just gives you a blank graphing space. Now here's the species. We've already plotted those before. Now imagine that our plots or sites, this time we called it communities, plot, site, or samples. We're divided into 2 treatments, and we want to display those on the graph. So this is like burned versus unburned prairie, for example. So first we're going to just say, hey, concatenate these treatment 1 and treatment 2, the first 5 treatment 1, the second 5 treatment 2. We're just going to overlay these, or just create a new variable, which is just treatment 1 and treatment 2. So type the code, run the code. Now we're going to graph some hulls. So we've got the, let's go ahead and get rid of this again. Let's create the graph space. Here it is. Display the species. I didn't need to do this. I already had this there. Oh, in this case, I made them gray, or Eric did. I think I did, actually. And then here's the code, this orditope to display the sites, like this. In this case, I made them blue and dark red. Here they are. And then we're going to draw a hull. The hulls connect. Here they are. The hulls connect all of those communities like that. So here's the code. Got it? I hope you've got it. So if you wanted to display your ordination and you said, okay, here are the species, here are the communities, and here's how they are displayed in space using this hull function. You could do it this way. That emphasizes the edge of the communities. Now, another way to do this is through, or a different approach other than the hull approach, is through ellipses, which I mentioned before in the PowerPoint. So let's draw some ellipses. So we draw the plot like this. That's a blank plot. It's got this type in. We are going to label the plot. It went over here for some reason. Here's the code. Species in gray. Communities in blue and maroon. That looks terrible. I can't tell what's going on. And then the ellipses code is this. So we're going to draw some ellipses, and this is the way this code is set up. So this is basically the code you want to run from here to here. Type the code, run the code. My drumming while you're coding. Got it? So this would be a good way to present, not good, common way to present data is like, oh, look, the communities overlap based on the overlap of these ellipses. Here's a way to draw ellipses on an ordination diagram. You can do it a different way, something quite similar. Let's draw the plot again. What I want to do, oops, let's get the labels down here. Connector lines. Sorry to do this in real time. Draw the plot. Label the plot, same as before. By the way, it's the same code. So you could actually have these things, this ellipses, ordi hull, and ordi spider. You could do that all just like on top of each other. This is copying and paste exercise. But the ordi spider command draws lines to connect them all. And this is really comes down to what your preference is as a data analyst and somebody who wants to present their thesis data. This is NMDS axis 1 and NMDS axis 2. In fact, we said K2, so there's only 2 axes in this, so you're good to go. Okay. This is it. And this last bit of code, I think, is just doing the same thing again we've already done in C. Yeah. Yeah, it doesn't even work. Okay, so this is not useful. Already plot. Label the plot. Draw the spider. Yep, that's good. So don't need any of this. Okay. Great. If you've got all that, then now I'm going to introduce you to, maybe take a break, pause. I'll just wait here. Okay, ready? So I'm going to introduce you to a new data set. You haven't seen this before. This is a data set from a paper that I wrote many years ago. It's an herb layer paper. Now, that is to say, it's a paper about forest herbs, forest plants. Let's see if I put it here. Yeah, I did. I'll share the PDF of this manuscript. It was published in 2011. And there's a lot of detail in this. I'm scrolling, sorry. Hopefully it's not nauseating. I'm going to scroll down. But one good thing about the reason I'm sharing this with you is there's a lot of detail in here about analysis techniques, including NMS. I call it NMS. I don't know, it's NMDS. One thing we did, and this hasn't been done with the data set we're going to work with today, is trimmed the data. So you might need to do that with your own data set. I'm saying I trim the matrix to include only species that occurred on 2 or more plots during any given sampling period. And then here's the sort of process I went through. It's explained in some detail here. So I'll share this with you. And we're not going to do all this today, but this is an example of NMDS. And if you find yourself running NMDS, you can use this as a way to think about how you should be doing it. Similarly, I've got plots, and this is the plot we're going to work on. We're going to basically work on this data set and make a plot similar to this one. It won't be identical, but it'll be somewhat similar to this one. We're not going to do the vectors today. The vectors would be a different exercise. It would just take more time, and I just don't think we can get to it. And then I'll also say that you can look in this, you can look at this results section and think about how you would present an NMDS in your own paper if you need to write an NMDS for your paper. So, ordination of the April sampling illustrated the importance of soil fertility and topography on herb community structure, figure 2. This is a good topic sentence in an ecology paper. This is ecological. I'm not talking about the results, PS. Doesn't say, it just says what's going on ecologically. The analysis 103 species, 74 plots included, explained a substantial portion of the total variation in the data set, R equals 0.6, R squared. Not surprisingly, given that the final stress was 20.9, final instability. These are things to report. We're going to look at stress in just a minute. I think our stress is going to be close to this. I did this not in R. R had not been, well, it wasn't a thing, I don't think, at that time. And then anyway, you can chase this down and see how I explain some of this stuff if you ever use NMS in your own research. So this is another example of like peer-reviewed journal article use of data analysis. And we can't chase it all the way down, but we'll get somewhat close. At least we'll play with the data set. So I will share this herbs data set with you. And so you'll read in the data, display the data. Now, here's the thing. I should go all the way across and do that again. This is a real biology data set. So what it means is, look, this is I'm scrolling the data set right here. I'm still scrolling it. So all the way, I mean, this is a huge data set. And I can show it to you in Excel right here, actually. I've got the data set in Excel right here. I'll share that too, just so you have it. But this gives you an idea of how deep it is, but it goes this way a long way. This is a species matrix. I have measured and found all those species in the plots. Now, one of the interesting things and thing you're going to have to ponder if you actually do NMDS in your own study is that these community matrices oftentimes have a lot of zeros. So this one, I think that's Trillium erectum, which is a wildflower. It's only found on 2 plots. That's not good. Here's Tiarella. Tiarella is found in some of the plots, but mostly not. It's only found on this set of plots right here. Tiarella is called foam flower. Tiarella cordifolia, I think. Anyway, you got to figure out what you're going to do about that. Are you going to trim those? How many plots do they have to be on? Because you have all of these zeros in your data set. This is a look at this one. This is a fern, I'm pretty sure. It's only on 1 plot. So actually, that should have, well, this is a full data set, but it's going to be trimmed. It wasn't trimmed in the paper, actually. So that species probably, I don't know. Anyway, I'm not sure where we are, but you might want to trim that out of your data set if you had that. Like that plant occurs on 1 of your 80 plots. Does it stay in? What do you do? I don't have an answer for you, but I do want to say, because some of you might actually do NMDS with your own data, you're going to have a rectangular data matrix with a lot of zeros. People have been working on that for a long time, and you should look into it. Think about carefully what you're going to do about that. Are you going to trim the data set or what? Okay, so we need to prep the matrix a little bit for the analysis, so we're going to do a subset here. We've talked about subsetting before already. So we did a subset from just columns 3 to columns 105, and now we can take a look. I just cut out the plot and community columns that were there before. Now we're going to rename the rows based on the plot names. So the plot names are now encapsulated in the rows, actually. So here, instead of it always, every time we've ever run R, it's just counted. Now we got the plot names there. So see if you can make this happen. Okay. Now let's run the NMDS. Here's running. Watch. I got an hourglass. It's the first time we've got an hourglass. Now it's saying 100 tries. This was try max is 100. If I was really doing this for research, it'd probably be 1,000. When I was in grad school, when we ran an NMDS, it was like over the weekend. It would be running and running and running and running. So computers are so much faster, of course, now, but infinitely faster almost. But you saw it run over there. And the final stress ended up being 14. I think Eric Borth somewhere said, and I've read this before too, less than 0.2 is okay. You're functional. Less than 0.1 is good. So that's fine. We're okay, is what I would say. I don't really know how to tell you what to tell you about the stress plot. I don't fully understand it. Like I said, I looked into it. It said the points are close to the line. It looks okay. The R squared seems good. If you're going to use it for research, I would say you need to figure out exactly what that's all about. That was not part of the thing that I did back in 2011 or whatever. So now let's draw it, or let's plot it. Here's the ordination diagram. Now we're going to label the plot. Oh, whoops. Let's go through the process like this. Draw the plot, ordi plot. label the species like this. I'll let you do it. So draw the plot, label the species. I won't drum anymore. I don't know if it's a terrible sound or what. I'll just be waiting. Once you've got that, we want to plot the sites. This is how you plot the sites. It's going to look like this. Whoa, complicated. Because there's a lot of sites. It's 80. I don't know how many are in the data set, actually, because it's like I said, it's been trimmed. But here's the code for that right here. Hopefully that worked for you. Now, our last thing to do is draw ellipses. Oh, sorry, let you do it first. There you go. Type the code, run the code. You'll see the ellipses pop up. There they are. I put labels on there. Put a label equals true. Okay, so here's what we've got. This is really weird. I'm going to code it. I'm going to run it again. Okay, hold on a second. Here we go. Okay. So now we've got these three communities, oak, beech, and mesophytic, displayed on this diagram using these ellipses. Now, I might not even have the plots on there. I might do it like this, actually, because I don't think I had the plot names on my other one. Just go species, and then straight to ellipses. See how that looks. That's a little better, a little bit more doable. Now, we're not nearly ready to send this to a journal for review, right? Look, if you're going to do ordination, you're going to take some time. It's going to be a month. It's going to be a month, probably, if you're going to get a really nice figure. Like this figure from my manuscript, this was not done in an afternoon, y'all. Okay, I spent a lot of time thinking about this. I didn't just crank it out. So you need to just invest that kind of time. But let's just take a quick side-by-side here if we can. Now, in my published NMS figure, I had NMS axis 1 and NMS axis 2, which we have here as well. Good. I have these triangles are oak. That's the oak community. So these are oak community. I don't have the points on here arrayed that way, and I'm sure we could, but we do have this ellipses. Is it ellipsis? How do you say it's singular for ellipses? Ellipse that's on this side. So it really does kind of match up with this oak community. We've got mesophytic right here. The mesophytic in this one is the gray triangles. The mesophytic community is right there. And we've got beech. Beech is this open circle. The beech community is here, but it's overlapping. There is definitely, see this? If you took the gray ones and these open circles, they're overlapping right here. They're overlapping right there. And also, look at this. The oak community comes down here and overlaps somewhat with the beech. So this is like these open circles and these triangles. We see that here also. And there's some separation right here between oak and mesophytic, and we see that right here. So while we're not done by any means with this ordination as it is right here in R, you can see that after, with very simple coding actually, and not too much manipulation of the data set, we've come to a basic representation of a published figure, a published ordination. And we've got a stress that was reasonable, it was 0.14 or something like that. So we're more or less good to go on this thing. We're moving. What I want to say now is that if you use NMDS, let me know if you're around campus and it's before you've graduated. I do have quite a lot of experience with it, so I can help you. Hopefully that this presentation has got you started down the path towards being able to use this technique. And let me also just say, this is the last time we will code together, which is very sad. Sorry we couldn't wrap up together in the classroom. However, I wish you good health, Sláinte, and I really hope it was useful, y'all. I hope it was useful. I did what I could to give you a good class. So take care.